Assalamu alaikum students, hope you are fine. Today we are going to study about antigens and antibody interactions. By the end of this lecture, you will be able to understand what are immune complexes and how are they formed, different properties of antigen-antibody interactions like affinity, avidity, and cross-reactivity, and how these interactions lead to providing immunity to pathogens, that is via neutralization, destruction, or elimination of pathogen. Let us start the lecture. Antigen and antibody interactions are the basis of a humoral or antibody-mediated immunity. So the interaction of an antigen and antibody is quite similar to the binding of enzyme with a substrate in a manner of lock and key, and both of them involve hydrophobic and ionic interaction. But this process is reversible. As you can see, an interaction of antigen with antibody result in formation of a complex which is called immune complex or antigen antibody complex, and this process is reversible. These antigen and antibodies reaction form the basis for detection of infectious diseases causing agent.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen.
The three basic features of antigen-antibody reactions are binding sites, the type of bonding, and type of reaction. Let us discuss them one by one. The binding of antigen and antibodies is in the form of a lock and key. So the antigen is the form of a key which fits into the lock of an antibody. So the key of the antigen is generally the epitope, and the lock is formed by the paratope of the antibody. For a reminder, a paratope is present on the Fab region or fragment of antigen binding sites, and it is made up of hypervariable regions. So this epitope fits into the paratope like a key into a lock, similar to that of an enzyme substrate reaction. So when we have a closer look at how these antigens and antibodies interact at the epitope and the paratope, we can see there are different types of non-covalent bonds involved, like hydrogen bonding, ionic bonding, hydrophobic interactions, and van der Waals interactions. As all of these bonds are non-covalent, so the process is generally reversible, as you can see here in this reaction. Antigen-antibody interactions are highly specific. That is because an individual antibody or a clone of that antibody can bind to only one specific antigenic determinant or epitope. That is, the antibodies are able to distinguish differences in the primary structure of antigen, and it is also able to differentiate between the isomeric form of an antigen and also between the secondary and tertiary structure of an antigen. Let's look at how these antibodies produce such amount of diversity to identify all these differences with the help of this video.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. And billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.